Hey guys, this is Mr. Mahmood, and you are watching Protein Synthesis Lecture 1. Now this is a pretty quick unit, so there are only going to be 2 lectures here, but that doesn't take away from the importance of what you're going to be learning in this unit. This unit discusses what's considered the central dogma of biology, which means everything that happens within biology rotates around the basic idea that DNA creates your proteins, and then those proteins perform the functions of your cell. Everything that revolves around biology bases itself on that in terms of how the cells work. So we're going to walk through the basic structure of that central dogma, which is a process known as protein synthesis. The 2 basic parts are called transcription and translation. But before we go into those 2 basic parts of protein synthesis, we're going to go back and remind ourselves of the 2 basic categories of nucleic acids, which are RNA and DNA. You already know quite a bit about DNA, but remember you were introduced to RNA back with biochemistry, and now it's going to play a leading role here once again. So we'll spend some time making sure you understand the basic differences between the 2 structures. We'll talk about 3 types of RNA that are going to be used in this overall process, and then we're going to jump right into protein synthesis itself. Alright, so let's get into a basic discussion of nucleic acids. We've spent a lot of time on DNA already. You guys should be very comfortable with the basic components of DNA, some of the characteristics about it. So you see the DNA line there on the right. DNA is double-stranded, that means it's 2 sides. It's a helical rotation, so that means it's rotated. You remember Chargaff's rules. There are 4 bases in DNA. The 2 purines are adenine and guanine. The 2 pyrimidines are cytosine and thymine. And they bond together with a very specific rule. A bonds to T, adenine bonds to thymine. C bonds to G, cytosine bonds to guanine. So if you know one side of the DNA line, you should be able to tell me the other very easily just using Chargaff's rule. Right? Basic stuff we talked about here. The human DNA line is about 3 billion base pairs long, very, very long in terms of everything that it has inside. It has to be long to hold all of that genetic information, right? Now let's talk about RNA in comparison to DNA. There's one particular type of RNA that we're going to be referring to here. This is specifically something called messenger RNA, which we'll get to later. But the basic differences between RNA and DNA is what you see here. Structurally, they're very similar because, in fact, RNA is made from DNA all the time. When we talk about cells functioning and making the protein, the process starts by an RNA copy being made from the DNA. So it would make sense that RNA is very much like the DNA itself because it's a copy of it. So it does have a lot of the same basic components, a lot of the same basic structures. It has a nucleotide with the same 3 basic parts, a sugar, a phosphate, and a base, but we'll talk about how those are a little bit different. Structurally, you'll notice one glaring difference here, right? DNA has 2 sides, it's double-stranded. Look at the RNA line. It's just one. It's single-stranded, and it's because it's only going to copy one side. So if you're making a copy of one side, you also will just be one side. So RNA is actually single-stranded, whereas DNA is double-stranded. That's one of the major differences between the 2. You're looking at the DNA bases, you notice 3 of the 4 are the same. Adenine is still present in RNA, guanine is still present in RNA, and cytosine is still present in RNA. However, there is no thymine. You might notice if you look at the RNA strand there on the left, there are no red rungs of that ladder because that red represents the thymine there on the DNA. So there are no thymines in RNA. So you may ask me, Mr. Mahmood, why does DNA have thymine but then RNA switches out to uracil? It's actually the other way around. So if you remember back when we were talking about early understandings of early life and their first organic compounds that were developed, the current theory, remember, is that RNA was the first organic compound present on Earth, and that it stemmed into all of the different components that we know today. So RNA, remember, theoretically actually came first. Even though within a cell's process here of protein synthesis, the DNA is the starting point, and you make RNA from it. If we think all the way back to early Earth, RNA actually came first. So the idea here is that those 4 bases, A, U, C, and G, were the first bases present on Earth in terms of organic compounds. So you may remember in that discussion, those scientists, the British scientists in the lab that made the first RNA molecule, that were able to make it in the laboratory, they made a cytosine nucleotide, and it was an RNA nucleotide. And then they exposed it to a little light, and that cytosine immediately switched over to uracil. That's something that can happen relatively easy inside of a cell. So if you were to leave an RNA line too long in the cell, the cytosines and the uracils could kind of get confused because over time those uracils could little by little start switching over to cytosines. So if you're really worried about the sequence of bases in terms of instructing your cells on what to do, if you mess up those sequence of bases because U's are switching over to C's, then you have a pretty serious problem. So over time, the long-term genetic material, which is your DNA, has advanced and evolved itself to no longer use uracil, but instead to use something very similar that we call thymine. That thymine base is much more structurally sound, and that means it won't switch over to cytosine nearly as easily as the RNA's uracil would have been able to. So if you're wondering why DNA has T's and RNA has U's, it's because the U's are really unstable, and if that U was in a long-term genetic line like DNA, it could very easily switch over to the other pyrimidine, which is a C, and that would completely mess up the sequence, and then in turn completely mess up the proteins that get made from it. So RNA has uracil, DNA has thymine. Because RNAs, as we're going to talk about here, are usually short-term genetic lines, they're only used for a short period of time, it's okay that you're talking about a relatively unstable base like uracil, because it usually doesn't have enough time to have any sort of a switch over like that until it's already been used up. And at that point it doesn't matter. And in terms of Chargaff's rule, it kind of works the same way. Everything's the same, except anywhere you normally put a thymine, if you're talking about any type of RNA, you would just put a uracil in its place. So in this case, A bonds to U and C bonds to G. So remember, RNA has no T's whatsoever. Uracils are what are used in their place. Another major difference comes in its location, as we're going to talk about here throughout the process of protein synthesis. You should already know, in a eukaryotic cell, the DNA is always found in the nucleus, right? So the DNA will always be in the nucleus of a eukaryotic cell and never leaves the nucleus. It's one of the major reasons the nucleus was developed, to protect the DNA. But as you're going to learn, the RNA starts in the nucleus because remember, it's made as a copy of the DNA, so it starts in the nucleus. However, it's actually going to be able to exit the nucleus, and it's going to go off and help put together and assemble the proteins out on the ribosomes, which is outside of that nucleus. So we've talked about 3 of the 4 major differences between DNA and RNA. So to talk about the 4th major difference between DNA and RNA, let's look at the actual detailed structure of the nucleotide. So if you look carefully here, both DNA and RNA have the same 3 basic parts of a nucleotide. They have a phosphate, a 5-carbon sugar, and then one of 4 nitrogen bases. We've already said how the 4 nitrogen bases are slightly different. 3 of the 4 are the same, but there is no thymine, and instead you have a uracil. And if you look at the structural components there, you can see the difference between the two. Both of these nucleotides represent the bases that we're talking about switching out. So in the RNA one, which is the blue pictured nucleotide there, that base that's there is a uracil. Compared to the DNA, the yellow line, that base is a thymine. So you see they're very similar to each other. The only difference comes in that single carbon that's up there on the top left. In the RNA line, the uracil, it doesn't have anything extending off of it. It just has a hydrogen to keep it stable. But in the DNA line, there's actually another carbon that's extending off of that. And that extra carbon gives it a lot more stability and is able to keep it from switching over to a cytosine the way that uracil could easily switch over. So the uracil is a little less stable, and that's why you see it only in RNA, not in DNA. But the major difference structurally in terms of the nucleotides between the two comes in the sugar. You should remember from our biochemistry unit that the D in DNA and the R in RNA are named after the sugar that's found in their nucleotides. We already know the D in DNA stands for deoxyribose, and we've explained this before. It's that second carbon that names this sugar. If you look at that second carbon, what's extending off of it is just a hydrogen, which means it's missing the oxygen that might normally be there, and therefore it's deoxyed. If you compare that to the RNA sugar, you'll see that's the oxygen that I'm referring to. That second carbon has an OH, or a hydroxyl group, instead of just an H. So because it has that O, it's no longer deoxyed. It has the oxy. So that means we now can just call this sugar ribose. So RNA nucleotide has ribose as its 5-carbon sugar, and that's where the name comes from. So RNA stands for ribonucleic acid, and DNA, again, because it's missing the O, stands for deoxyribonucleic acid. So that's the 4th major difference between RNA and DNA. RNA has the sugar ribose, whereas DNA has the sugar deoxyribose. So make sure you know the 4 major differences between DNA and RNA. And when we talk about RNA in this example, we're always referring to a type of RNA known as messenger RNA. The messenger RNA, or known as mRNA, is the one that's the direct copy that's taken from the nucleus. I made a reference in a previous unit to the DNA being more of the long-term template for the cell. So it would be like if you were an architect and you hand drew this layout of this building, a blueprint of this building. You don't want to take that blueprint out of your office because you don't want anything to happen to that blueprint because all of the work you put into it. So instead you make a copy of the blueprint, and that copy is what's able to go out onto the site, into the construction site, and that's where you can build on to make your actual building. And if anything were to happen to the copy, it's okay because you could always come back to the original that's protected in your office. That's basically how DNA and RNA work here. So that messenger, the mRNA, is the copy that's going to be made from the nucleus that's going to actually be able to exit the nucleus and then be a major part to help put together the correct protein based on what that original DNA had wanted. So this mRNA is the one that's going to be the copy of the DNA, but it's also going to need to connect itself with the parts of a protein. So the mRNA is going to bring the information from the DNA that knows the sequence that the protein is going to need to be in, but it's the tRNA, also known as transfer RNA, that's going to actually be able to connect the sequence of the mRNA with the correct amino acids in the right order that's going to actually make the protein. The mRNA has the sequence from the DNA, so it knows what the sequence should be, but it's going to be the tRNA that actually brings the amino acids. So the tRNA is going to come in, match up with the mRNA, and we'll talk about how, and by matching up in a certain place, it'll put its amino acid in that position as well. If you look at this tRNA structure, again, we'll talk more about this later, it has two distinct parts on either side. First off, it looks like a T, so that should, if you're ever confused on a test or a quiz, and you see a structure and you're trying to identify the types, if it looks like a T like this, it is T, it's the tRNA. So on one side of the tRNA has a 3-base sequence that you see there at the bottom. That 3-base sequence is what's going to be used to match up to the right spot on the messenger RNA. That's how they go to the right place. And they use the same corresponding base pair rules that we've talked about before. And then on the other side of that tRNA is an actual amino acid. And as we'll talk about this again, those amino acids are what are going to combine together using peptide bonds to make your polypeptide or your protein. So the mRNA knows the sequence, the tRNA actually brings the amino acids to be put in the right sequence. And then the third type of RNA is known as ribosomal RNA, also known as rRNA, and it's what makes up the actual ribosome, and it'll help make sure that these two parts, the messenger RNA and the transfer RNA, are matching up correctly in order to put together the correct protein. And remember, the ribosome is the assembly site where these proteins are actually going to be put together. So all of this happens through a process known as protein synthesis. So the whole process pretty much takes place in one of two locations. In the nucleus, the focus is going to be on getting that messenger RNA made from the DNA. The first part in the nucleus is known as transcription. To transcribe something literally means to make a copy. So transcription is the whole process of making that mRNA copy from the DNA. And then once the mRNA is made and exits the nucleus, we now go into the second half of protein synthesis occurring out on the ribosome in the cytoplasm, which is called translation. And as you know, translating literally means to go from one language to the next, and that's what's going to be happening here. We're going to go from the language of bases, which is your original DNA line that was now copied into the RNA, so the sequence of bases is one language, and then the translated language that's going to match up to those bases is going to be the amino acids. So you're literally translating from bases to amino acids so that you can now make a protein from that original DNA line. And that second half is called translation. It's what happens out on the ribosome. Alright, so let's go through each one very specifically. So you'll notice transcription is a whole lot like DNA replication. It has a lot of the same components, very similar enzymes, if not the exact same enzymes, and a lot of the same steps. But there's some key differences here. So you remember, just a quick review of DNA replication. You start with one DNA line and you end with two identical copies of the same DNA. And that happens because when the DNA unzips, you're copying both sides of the DNA. Remember, not in the same direction. One starts from the top, the other starts from the bottom, but you're eventually going to copy out both sides of the DNA, putting in new nucleotides in the middle, so you end up with two identical strands. This process of transcription starts out the exact same way. So we begin with the exact same enzyme performing the exact same job. You remember, there is one particular type of bond in this whole structure that's weaker than the others, and that's a good sign because it's actually what's needed to break apart. And those are the hydrogen bonds that are sitting in between the bases, right? Remember, A and T have 2 hydrogen bonds holding them together. G's and C's have 3 hydrogen bonds holding them together. So the first step in both DNA replication and in transcription is the need to first unzip the DNA so that you have room in the middle to actually start making any kind of a copy at all. And what's the name of that enzyme? Helicase. Good. So DNA helicase is going to go through and unzip the DNA. So it's going to break all of the hydrogen bonds that are within that DNA line, and that'll allow the DNA to unzip or to separate. Now, the key thing to remember here is we're now talking about just getting a copy of a certain section of the DNA. The human DNA line has about 30,000 genes. It's made up of about 30,000 genes. And a gene is any segment of the DNA line responsible for producing one protein. So any particular gene is the DNA sequence that will in turn put together one particular type of protein. So when we're talking about DNA replication, you eventually have to unzip the entire DNA line because you're needing to make 2 identical copies that can split into 2 completely different cells from each other and function independently. However, when we're talking about transcription, the goal is just one step at a time. There's no need to copy the whole DNA line every single time because the goal here is just to make one particular protein. So when we talk about this first step with DNA helicase, even though it's the same basic process, it doesn't unzip the entire DNA. Instead, it just unzips a certain section, just enough for that particular protein, which means it'll only unzip one gene. Helicase unzips the entire DNA if it's DNA replication, but in this case this is just transcription. It's only going to unzip one gene. It's only going to unzip one segment of the DNA just long enough to be able to pull it and use it to put together one particular protein. So the DNA unzips. It's the same basic first step, it's just not the whole line. Helicase unzips the DNA. The second step, remember, in DNA replication was that you brought in an enzyme to attract DNA nucleotides to the area. That enzyme before was called DNA polymerase. However, keep in mind, this time we're not making DNA. This time we're making RNA. So we're still going to have a polymerase enzyme that will help bring in the nucleotides needed to put together your RNA, but this time instead of DNA polymerase, it's actually called RNA polymerase. So this time RNA polymerase is going to bring RNA nucleotides to the site. And remember the basic differences between an RNA nucleotide. All RNA nucleotides have a different sugar. So in this case, instead of that 5-carbon sugar being deoxyribose, now that 5-carbon sugar is ribose, right? And with the exception of only one of the bases, everything else is the same. So here, if I have a thymine on that 3' end there, on the right side, what's it going to bond to in terms of RNA? Just like DNA, it's the same thing. If I have a thymine on one side, I have an adenine on the other. Adenine's base is perfectly comfortable in both DNA and RNA. So if I have a T on the DNA line, it would match up to an A in RNA, just like it would if it was DNA. So an A RNA nucleotide will come in and match up. The second sequence, if you look at that base down there at the bottom, it's labeled as a guanine on the left side. So I have a guanine there on the right side. What RNA nucleotide is going to be attracted there? A C, a cytosine, right? C and G are the exact same in DNA and RNA. There's no difference. They match up to each other. So this cytosine will match up, except again the sugar is different. It's ribose now instead of deoxyribose. The next sequence, this is the one that you have to be careful for. If you look down there on the right, I have an adenine. So if I have an adenine on the right, if you look at the original DNA line on the left, there's a thymine there because A bonds to T. But in RNA, there are no thymines. So what nucleotide is going to come in this place to match up with that adenine? This is going to be that uracil, exactly. And I know they look the same here, but uracil is what's going to be the RNA nucleotide that's going to match up here. So this nucleotide not only has a different 5-carbon sugar, but it has a different base from the DNA line. So this is the uracil that's going to match up to an A. And then the last one, it looks like I have a C or a cytosine at that bottom right. So I'd have a guanine nucleotide coming in and matching up. So this second step, again, is very similar to DNA replication. A polymerase brings in nucleotides, except this time it's RNA polymerase that brings in RNA nucleotides. They match up to the corresponding bases. Remember, you have 4 bases of RNA: adenine, uracil, cytosine, and guanine. So make sure you only use those 4 bases. Anywhere you would normally have put a thymine, just make sure you switch it out with a uracil. So if you see a T, it bonds to an A, whether I'm talking about DNA or RNA, it doesn't matter. However, if you see an A on the DNA line, you have to be careful. If I'm asking about the other side of the DNA, it would be a T. But if I'm asking about the RNA that would match up to that A, it would be a uracil, or a U. So just remember, no T's in RNA. So if we were thinking about DNA replication, this process would now continue on the right side. And you would see the exact opposite thing happening on the right side. You'd be matching up those bases as well. But remember, I'm not trying to make extra DNA here. My goal is simply to copy one side of the DNA line, and I've already done that. So now the third step that's going to be used here, remember that enzyme that's going to help glue all the bonds together and form those bonds properly, is ligase. The ligase before was focused on helping put those hydrogen bonds together in the middle to put the DNA structure together. In this case, that ligase is going to help to make sure that each of the individual nucleotides are bonded properly to each other, that each phosphate is bonded to the sugar above it so that they're matched together, so that I can have a fully structured single-stranded RNA line. Once I have all parts made, that ligase has sort of glued all the parts together, made sure the bonds are proper, then this RNA line will separate off of the DNA that it was copying from, and you now have created a messenger RNA line. So this is our mRNA, and now at the end of transcription here, this mRNA will actually be able to leave the nucleus and start its track out of the nucleus, out into the cytoplasm, and eventually to the ribosome. And then the DNA line here is going to go right back to normal. So it's been unzipped, and now the final step is that DNA is going to rezip or reform, and once again we have that ligase that's going to come in and reform those hydrogen bonds so that everything is back to normal in terms of DNA. So we started with one DNA line and we end with one DNA line. There's no copying of DNA that's happening here. There's no replication here. This is purely for the sake of making a protein. So I just unzip the DNA, copy a segment just long enough for one protein, which is one gene, and I make that mRNA line. That mRNA breaks off of that one side, leaves out to start making the protein, and then the DNA just closes right back up. So it's just like a textbook or something like that. If you need a copy of page 247, you can make that copy as many times as you want, but you always end up with the same book that only just has one page 247. You're just using the same book over and over again. Alright, so now that we have the messenger RNA, we have the base sequence that's going to determine which amino acids match up in which order to actually make the correct protein. We can go into the second half of protein synthesis, which is known as translation. So the messenger RNA is represented here on the left. It's making its trek, and it's eventually going to make its way to the ribosome, like you see here. And it's going to kind of zip its way through. A ribosome has two subunits. If you look there, there's the red part and the blue part. What's known as the large and the small subunit. The messenger RNA is just going to sort of slide in between. And as it's sliding in between, that's where it's kind of positioning itself and making it ready to match up with the other type of RNA that we're going to be talking about here, which is the tRNA. So as the mRNA is going to be kind of sliding through the ribosome, it's going to be matching up one by one with its corresponding transfer RNA. The way that they know how to match up is based purely on the bases that are on both the messenger RNA and on the transfer RNA. So the messenger RNA is now going to be this little squiggly line there at the bottom, the blue line, but it again represents everything that we just saw. So the messenger RNA is nothing but a single-stranded line that's an exact copy of one side of the DNA. So it has a very specific sequence of bases, and those bases are any categories of A's, U's, C's, and G's. Now the tRNA on the bottom side has its own 3-base sequence. So because of its 3-base sequence, it's going to know where to match up on the messenger RNA. And it's all because of the bases. If I have a very unique sequence, for example, the example I have of the tRNA is UGG. Its 3-base sequence there at the bottom is UGG. So that UGG is going to kind of move around in that area, waiting for its corresponding bases to show up on the mRNA. So if the tRNA's bases are UGG, the mRNA's bases would be the exact opposite. So in this case, as you can see on the line, they would be ACC. So when an ACC triplet shows up, when the 3-base sequence of ACC shows up in the mRNA, that's where that tRNA is going to jump in. It naturally will match up because those bases match up to each other. So the last thing we need to talk about here is some of the terminology that we see here in terms of the bases. Every 3-base sequence on the messenger RNA has a name. Every 3 bases of the messenger RNA is called a codon, C-O-D-O-N. A codon is the 3-base sequence on RNA. Why is it 3 bases? Why not 2? Why not 4? The answer is because of the tRNA. Your tRNA always has a 3-base sequence that's trying to match up to the codon, and it's always the exact opposite of the codon. That's how they know how to match up. So these 3 bases on the tRNA fittingly are called anticodons. So an anticodon is the exact opposite of a codon, and that's how they match up. So the codons are the 3-base sequences on the mRNA, the anticodons are the 3-base sequences on the tRNA, and they match up to each other, bringing the amino acids in the right place. This process is in no way random. In fact, it's very much calculated, and you can make very specific predictions about the types of amino acids that will get put together because of their base sequences. They're very specific. Any kind of a particular amino acid only attaches to a certain type of tRNA that has a very specific anticodon. So if you know the base sequence of the DNA, then you can figure out the base sequence of your messenger RNA, or the copy, which would just be corresponding bases with no T's, right? And if you know that messenger RNA line, scientists have actually put together a chart that you can use to be able to predict the amino acid that would match up to that particular codon. And that is what we consider a codon chart, which you see here on the bottom right. So again, this visual here on the left shows you how every 3 bases of the messenger RNA represents a particular codon. They're not separated from each other physically in any way in the line, but because every tRNA's anticodon has 3 bases, each anticodon matches up to 3 bases on the messenger. So every 3 bases on the messenger is called the codon. So as the messenger RNA slides its way through the ribosome between the subunits, every time a codon pops up in a specific space right in the middle of that ribosome, the anticodons will look to find the matching one. And the matching anticodon will come in that place, put that amino acid in the order. Then it zips over. Then the next 3-base codon opens up, the next 3-base anticodon matches up to it, and that amino acid lines up next to it. Then the next codon continues, and the same process happens over and over again, putting all the amino acids in the right order. Another thing that's important to make sure you understand, as the mRNA starts to pass its way through, it's not just that first 3-base sequence that passes in the line that just automatically connects with an anticodon. In fact, the messenger RNA line will pass through the ribosome pretty far, usually, until it hits one particular codon segment. There's a specific codon segment, a 3-base sequence known as AUG in that mRNA copy, adenine, uracil, and guanine. If AUG is in that 3-base sequence, only then will the first amino acid actually start to attach. So you can see a diagram here that represents that. There are certain parts of that messenger RNA line that are known as UTRs, untranslated regions, because they're parts that don't have that AUG. So if you don't have that AUG anywhere, it's just going to continue to pass through until it hits that AUG line. That AUG messenger RNA codon is known as the start codon. It has a very specific name. So the start codon is that first codon that's going to actually attract a tRNA, which means it actually has with it a particular amino acid. So anytime we're thinking about a start codon, there's only one. It's called AUG. The amino acid that it attracts is called methionine. Now there are 20 different amino acids. You've learned this already. There are 20 different amino acids that make up proteins. 12 of them are naturally made by the body. 8 of them you have to get through your diet. That's why they're called essential amino acids. And we said before, it is very important that you always have all 20 amino acids available at any given time, so you're putting together the correct protein. This is where it all happens. So to know which amino acids are going to be coming in and matching up in any given codon, you use this chart that's down here on the bottom right. It's very straightforward, very basic. If you learn it the first time, hopefully you'll know how to use it every time after that. If you know what the messenger RNA codon is, then all you have to do is look at this chart to figure out which amino acid would match up. And it's very simple. The 3 bases of the codon represent 3 sides of the chart. So you always start with the first side, which is on the left. So you're going to start with the left side vertically of the chart with the first base of your codon. The second base will represent the top of the chart, and the third base will represent the right side of the chart. So basically, your first base is going to tell you which of the 4 rows you're going to be focusing on here. So you'll notice there are 4 rows on this chart. There's what's considered a U row, a C row, an A row, and then a G row. Notice how there are no thymines here because all of this is RNA, right? So in the messenger RNA line, the first base tells you which of those 4 rows you're going to focus in on. The second base tells you which of the 4 columns you're going to focus in on. Again, there's a U column, a C column, an A column, and a G column. So if, for example, we start with that very important start codon that we've talked about before, which is AUG. A, for the first base, would tell me I'm focusing on the 3rd row of the chart. So AUG. So the second base is U. So that tells me of the 4 columns, all I care about is the first column. Where the 2 intersect is going to be the box that I'm going to focus in on. And then within that box, there are 4 possible lines there. And a lot of times they have the same ones in more than one place, but there are 4 possible segments of that box. And that's where the third base comes in. The third base will tell me within that box that intersects between the column and the row, within that box, I want to know which of the 4 I really need to focus in on. And that's where the third base comes in. So in this case, AUG would tell me I'm worried about the 3rd row, first column, and then within that box particularly, I'm looking at the very last option of that box, which in this case, as you can see, says start, or also methionine. So the start codon, which is AUG, will always attract a methionine amino acid, which is the beginning of every single protein of your body. Every protein you make begins with a methionine amino acid because that's what's attracted to your start codon. And once you've hit that methionine, from that point on, every other amino acid that comes in is going to bond with the previous. And we're going to keep going until we reach something known as a stop codon, which you may be able to find already in the chart. But let's not get ahead of ourselves. Let's do some practice here. So we're going to use the RNA line there that's on the left side, and we're going to use those 7 codon sequences to predict the 7 amino acids that would match up to it. So the first codon is AUG. So we've already done that matchup to identify that as a methionine. So AUG is our start codon, so that begins our protein. So we're going to begin with a methionine. The second codon sequence is ACG. So take a minute and tell me which amino acid would match up to ACG. I hope you got threonine, right? It's threonine because it's the A and the C tells me I'm only worried about the 3rd row and the 2nd column. And all 4 of those are threonine, so it really doesn't matter what the third base in this codon sequence is because they're all going to read for threonine. But just for practice sake, it's ACG, so it would be the last threonine in the box there officially. That would be the one I'm representing. Codon 3 is GAG. What would that match up to? It says glutamate there. It's also known as glutamic acid. Either way, it's fine. So we'll just go with the chart. Glutamate is 3rd. CUU, what would be the amino acid CUU would match to? Hopefully you got a leucine, right? The next one is CGG, codon number 5. What would be the amino acid that CGG would match to? Arginine, good. And then codon 6, AGC, would match to what? A serine, good. And then our final codon, codon 7, is UAG. What amino acid would that match to? Ah, you see that one says stop. Stop is not an amino acid, right? They didn't run out of names and just go with something that they already knew. Stop doesn't actually represent an amino acid. Stop represents the termination of your protein. So once you hit a stop codon, there is no amino acid there. So the amino acid that was before it is now the end of the protein. So that's how your cells know when to stop making a protein, not to just go on all the way to the end of the chain. So once you hit a stop codon, then the amino acids that were before it, each one of which has bonded together, forming a peptide bond through dehydration synthesis, and is now going to be your protein, is going to break off, go through its folds and formation, jump into the Golgi apparatus to help properly make the forms and all of the stuff we've talked about before, and now you have your fully functional proteins ready to go out and perform the functions of the cell. So again, if you know the original DNA line, you can turn around and tell me what the mRNA copy would be if you're just copying one side. Then that mRNA copy, if you know that, you can turn around and tell me what the tRNA anticodons that would match up to the mRNA codons would be. Or you can jump straight to the codon chart if you know your messenger RNA line and be able to predict the amino acids that would match up to each individual codon. And now that sequence of amino acids would make for a very particular protein. And this is basically how your cells function. You start with the DNA, you end with the protein. So this is what we consider the central dogma of DNA. Just about everything within this concept of biology starts with DNA making RNAs, messenger RNA in particular, that go off to help assemble the proteins with the help of transfer RNA to turn around and perform the actual functions of the cell. If I'm talking about just DNA replication, there's the cyclical process that happens in the S phase before the cell jumps into mitosis. That way we're just talking about DNA making copies of itself. There's no RNA involved in that at all. However, if we're talking about the process of making proteins, we have to first go through transcription, which involves RNA polymerase, this time instead of DNA polymerase. So the messenger RNA that gets made goes out and with the help of tRNA and rRNA put together the correct sequence of amino acids to make the proper protein that'll then turn around and perform the functions of the cell. But basically most living things follow this central dogma, so it's very important you understand. Alright guys, I hope you got it all. Take care of yourselves and I'll see you next time.